Good evening. Am I audible and visible? If I'm audible and visible, can I see a quick thumbs up, please? Yeah. So, if you're joining my session for the first time, this is Dr. Shanmugapriya. I'm Professor and Head of the Department of Biochemistry at Government Thoothukudi Medical College. And I'm your Unacademy educator for Biochemistry. And today's session is an image-based MCQ discussion session on biochemistry. And I've chosen few MCQs related to genetics, few MCQs related to disorders, and few graph-based MCQs. Okay. So using these MCQs, I'll help you understand few concepts, and I will help you answer. I will help you to develop the ability to answer image-based MCQs. So that's an introduction to this session. And before we start the session, let me tell you few facts about Unacademy's plans. So today you have the last opportunity to get the Iconic subscription. As you all know, Iconic subscription is about subscription for both live Unacademy sessions and Prep Ladder. Because a recorded session on Unacademy platform has been released, today is the last date for you to get this Iconic subscription at an offer price. If you want 6 months subscription, the current price is 28,611, and for 24 month subscription, this is the cost. And for you to get this offer, you can use any educator's code. You can use my code, which is C Shanmugapriya. Yeah, 1 second. Yeah. Hi, Jay. Good evening. NEET PG is postponed. Hi, Jay. Good evening. So that's about Iconic subscription plan. And if you are an FMGE aspirant, we have Crack FMGE 2023 with FMGE Mastery, wherein all 19 subjects will be discussed by top educators. And we have 2 modules for this. The first one is all 19 subjects will be discussed with theory as a backup. And this will be between February and April. And second module will be a MCQ discussion session. So here we'll be discussing previous year questions and image-based questions. So these are the, so that's about FMG. Now about next 2023 batch. If you're a next 2023 aspirant, then we have Aspire 2023 batch, and the duration of this batch is 8 months. And in this batch, all sessions will be oriented towards next pattern. So mostly clinical case-based MCQs, image-based MCQs, conceptual MCQs will be discussed in this series. And these are the educators of this Aspire batch. And for NEET PG 2023, the last month preparation, we have a final booster batch, and the modules would include previous year question discussion, wherein all 19 subjects PYQs will be discussed and most expected MCQs will be discussed. And apart from that, you'll also have an access to BTR series by Dr. Zainab. So these are the offers that Unacademy has got currently. So now let's start discussing. Good evening, Shreya Patel. Good evening, Prabhakaran. Yeah, good day, Kwame Bempong. Okay, so can we start discussing this MCQ? So this is the first MCQ for today. Based on the image provided, which of the following is not a heterochromatin? Or in other words, they are asking you which of the following is a euchromatin. So do you have any idea what is a euchromatin and what is a heterochromatin? To tell you in simpler terms, so can you all try answering? Prabhakaran, Shreya Patel, Kwame, J. Munot. Anyone? What is euchromatin and what is heterochromatin? So to tell you in simpler terms, euchromatin is something which is transcriptionally active. And when it is transcriptionally active, it will be uncondensed. When it is uncondensed, it will stain less densely. So of all these parts, do you see that A is more densely stained? right? A is more densely stained, B is more densely stained, and then D is also more densely stained. So which is lightly stained? C is lightly stained. And what is C? C will be the arms of chromosome. So arms of chromosome will come under euchromatin type. Okay, so answer for this MCQ is C. Okay, answer for this MCQ is C. But for you to understand what is very good, all of you. Good, Dinesh Kumar. Hi, good evening, Dinesh Kumar. Yes, Vishnu, you're all right. Good evening, Shruzana. So let me tell you what is euchromatin and what is heterochromatin. And before we start discussing that, I want you to know about condensation of chromosome. First, tell me what is a chromosome. Any idea what is a chromosome? Yeah, chromosome is nothing but a long double-stranded DNA which has got condensed with the help of proteins. Don't we know that? Every chromosome, the one chromosome which you draw with the centromere, a short arm and a long arm, is nothing but one long double-stranded DNA which has got condensed with the help of proteins. And if you're asking me why should you condense chromosomes, why should you condense a long double-stranded DNA? It is because if you uncondense chromosome, every chromosome will measure a few meters long. And you will not be able to pack such a long double-stranded DNA within a nucleus which is a few microns in diameter. And that is why we condense chromosomes, we pack it inside nucleus which is a few microns in diameter. But as long as they are condensed, will they be able to perform their functions? No. So only as long as your chromosomes need not perform their functions of replication and transcription, your chromosomes will be condensed. Okay, so have a look at this. What is one chromosome? Every chromosome is a long double-stranded DNA which has got condensed with the help of proteins, and they remain condensed as long as they don't have to perform their functions of replication and transcription. But the moment they have to perform either replication or transcription, I hope you all know that for both replication and transcription there should be unwinding. And for unwinding, prior to that, there should be uncondensation. So whenever they have to perform their functions of replication or transcription, they undergo uncondensation. So this is an introduction. Now what are the types of chromatin? One second. So what are the types of chromatin? Chromatin is of two types. DNA wrapped along a tubular protein. Yes, proteins are called as histones, but not just histones, we have many other types of proteins which help in condensing chromosome, but of course histone is one of those proteins. Okay, so what are the types of chromatin? So the two types of chromatin are euchromatin and heterochromatin. Okay, euchromatin and heterochromatin. What is euchromatin? Euchromatin is transcriptionally active region of a chromosome. When I say it is transcriptionally active, how will it be? Will it be condensed or uncondensed? Just now I told you, right? When it is transcriptionally active, it has to be uncondensed. One second. Yeah, it has to be uncondensed. When it is uncondensed, how will it stain? It will stain less densely. In contrast, what is heterochromatin? Heterochromatin is transcriptionally inactive region of a chromosome. Yeah, it is transcriptionally inactive region of a chromosome. When it is transcriptionally inactive, how will it be? It will be highly condensed. When it is highly condensed, how will it stain? It will stain more densely. So that is about types of chromatin. So what are the two types of chromatin? Euchromatin and heterochromatin. Euchromatin is transcriptionally active region of a chromosome. When it is active, it is uncondensed, it stains less densely. Okay, whereas heterochromatin is highly condensed. Now what are the types of heterochromatin? The two types of heterochromatin are constitutive heterochromatin and facultative heterochromatin. So what are the two types of heterochromatin? One is constitutive heterochromatin, the other one is facultative heterochromatin. One second. There is some problem here. One second. So what is constitutive heterochromatin? Please explain porphyria at last. Yes, I will do it. I will do it. Hi Zainab. So what was I telling you? I was telling you constitutive heterochromatin is something which is transcriptionally inactive always. So where in your chromosome will you find something which is transcriptionally inactive always? It is found in the centromeric region and in the telomeric ends. And if you're asking me why will you want constitutive heterochromatin in the centromeric region, it is because, do you remember that in mitosis, mitotic spindle gets attached to the centromere, and in anaphase, when the mitotic spindle contracts, centromere breaks longitudinally. So centromere is a region which is subjected to repeated breakage. Okay, and whenever there is breakage, there will always be a tendency for one daughter chromosome to gain some of the sequences. There will be a tendency for the other daughter chromosome to miss some of the sequences. So tell me, what would have happened if nature had provided your centromere with a coding sequence? As a result of this, if there was a coding sequence in the centromeric region, following every cell division, one daughter cell would have undergone duplication of gene sequences, the other one would have undergone deletion of gene sequences, both of which are detrimental. This nature knows, and that is why chromosomes in the centromeric region, they are provided with constitutive heterochromatin, something which is inactive always. Okay, and then about telomeric ends. Why will you want such a heterochromatin in the telomeric ends? Because you must have learnt in replication that following every replication, telomeric end will undergo progressive shortening. So tell me, what would have happened if telomeric ends are provided with coding sequences? Again, following every cell division, the daughter cells would have undergone deletion of gene sequences, which is again detrimental. That is why along the centromeric regions and along the telomeric ends, you have constitutive heterochromatin, which is transcriptionally inactive always. And that is why you mark your centromere as a dark dot. Yeah, dark dot to denote that it will stain more densely, to tell you that it is highly condensed, to tell you that it is heterochromatin. Similarly, telomeric ends. Now, what is facultative heterochromatin? Facultative, as always, is it is sometimes active, sometimes inactive. Okay, so what will be an example of facultative heterochromatin? The best example will be female X chromosome. Okay, the best example will be female X chromosome. I hope you all know that all somatic cells of females have 2 X chromosomes. Though we have 2 X chromosomes, only one of the 2 X chromosomes will be transcriptionally active. And that is what is drawn with the centromere, a short arm and a long arm. Whereas the other X chromosome is transcriptionally inactive. And because it is transcriptionally inactive, you will mark it as a dark dot. And what do you call it as? You call it as a bar body. Yeah, that is called as, yes, I will start discussing it quickly. Okay, so that is called as bar body. So what is bar body? It is an example of facultative heterochromatin. So that's all about types of chromatin. Now try to answer this question. So the question is this. Yeah. So what is A? Just start answering. What is A? A is a telomeric end, right? A is telomeric end. What is B? B is centromere. Both A and B, yeah, both A and B are constitutive heterochromatin. Now look at D. What is D? D here is nothing but bar body, yeah, that is facultative heterochromatin. Whereas what is C? C are the arms of chromosome that will have euchromatin. So which of the following is not a heterochromatin, or which of the following is a euchromatin? Euchromatin is arms of the chromosome. Got it? Yes. So this is the next MCQ. Which of the following is a true statement? Try answering this. So this is the image that is provided to you. In this image, they have told you that this is G0 phase, okay? And then they have marked it as A, B, C, and finally M phase. So what is A, B, and C? Do you know that phases of cell cycle would include G0, G1, S, G2 and M phase. So what are the phases of cell cycle? G0, G1, S, G2 and M phases. Good evening, Dr. Sarika. So A is G1 phase and B is S phase. Yeah, with this understanding, now look at these choices. Which of the following is a true statement? Does replication happen in B phase? Where does replication happen? Replication happens in S phase. S phase or synthetic phase is so called because in this phase you synthesize two double-stranded DNAs from a single double-stranded DNA. So we know that replication happens in B phase. But when your chromosomes have to replicate, should the chromosome be condensed or uncondensed? Tell me. This is not good, ma'am. You sit and teach. Slides are not clear. Okay, next session I will sit and teach, Zainab. Now please try to follow this. Okay, try to follow this for this session. Okay, so replication happens in B phase. Okay, that far we are sure. But how will the chromosome be? Will the chromosome be condensed or uncondensed? For replication to happen, I said there should be first uncondensation followed by unwinding. So the answer here is replication happens in B phase, but in B phase the chromosomes are highly condensed is wrong. So replication happens in B phase, and in B phase the chromosomes will be uncondensed. So the right answer here is choice C. Okay, the right answer here is choice C. Now let's try to understand phases of cell cycle. Okay, I'm going to quickly tell you about phases of cell cycle. What are the phases of cell cycle? It starts with G0 phase. Yeah, it starts with G0 phase, that is followed by G1, S, G2 and M phase. So tell me what happens in M phase. In M phase, one cell divides into two. Don't you know that? In M phase, one cell divides into two. And after a cell divides into two, the two daughter cells either go to the complete resting phase or they get ready and they get into the next cell cycle. Yeah, after cell division, the two daughter cells either go to complete resting phase or they get ready for the next cell cycle. So this completes a cell cycle. Okay. Now what is G0 phase? G0 phase is a complete resting phase wherein neither replication nor transcription happens. Don't we know that? G0 is a complete resting phase wherein neither replication nor transcription happens. And when your cells need not perform replication and transcription, how should the chromosome be? The chromosome has to be highly condensed. So how do you want your chromosomes to be in G0 phase? Your chromosomes have to be highly condensed. And that is why a cell in G0 phase is drawn like this. So do you see that in this cell, there are two pairs of chromosome. This is one pair, this is the next pair. So there are two pairs of chromosomes and both the pairs of chromosomes are highly condensed. This is how your chromosomes will exist in G0 phase. Now in M phase, how do you want the chromosomes to be? Do you know that in M phase, just now I told you, in M phase, metaphase, mitotic spindle is formed, it gets attached to the centromere. And in anaphase, when the mitotic spindle contracts, the centromere breaks longitudinally. Which means in M phase, you want your chromosomes to break and segregate. And for breakage and segregation to happen, what form of chromosome do you want? Only when the chromosome is highly condensed and only when the chromosome is attached at one discrete point, breakage and segregation will be easier. Okay, so in M phase, because you want breakage and segregation to happen, how do you want the chromosome to be? The chromosome here should be again condensed. Can you see that here? The chromosome even here will be condensed. But how will you draw the chromosome in M phase? In M phase, you are going to draw the chromosome like this. So can you have a look at these two images and tell me the difference? What is the difference between chromosomes that exist in G0 phase and the chromosomes that exist in M phase? Do you see that in M phase, every chromosome has been duplicated? All of you, do you see that in M phase, every chromosome, thank you, Dr. Deeksha. Yeah, it has been duplicated. Every chromosome has been duplicated. When do you think this duplication happens? Duplication of chromosome or replication happens in S phase. So S phase or synthetic phase is so called because in this phase you synthesize two double-stranded DNAs from a single double-stranded DNA. And for replication to happen, what form of chromosome do you want? For replication, there should be uncondensation followed by unwinding. So in S phase, you want your chromosomes to be uncondensed. Okay. Now what is G1 phase? Give a logical name to G1 phase. G1 phase is a pre-synthetic phase. What is done in pre-synthetic phase? In pre-synthetic phase, the stage is set in such a way that replication can happen in S phase. You cannot randomly duplicate every chromosome that enters into the cell cycle. Before you duplicate the chromosome, you should proofread the chromosome. If there is any error, you have to repair it and then you push it into replication. So what is done in G1 phase? In G1 phase, we proofread and repair chromosomes. For proofreading and repair, what form of chromosome do you want? For proofreading and repair enzymes to be able to go and act on the chromosome has to be uncondensed. So not only in S phase, even in G1 phase, you want your chromosomes to be uncondensed. Now what is G2 phase? Give a logical name to G2 phase. If G1 phase is pre-synthetic phase, G2 phase will be a post-synthetic phase or a pre-mitotic phase, wherein you double check the daughter DNAs. So in S phase, you know, replication would have happened. So every chromosome will have 2 long double-stranded DNAs. But even during replication, you might have introduced defects. So before you allow the cells to divide, you have to proofread and repair those chromosomes. So again in G2 phase, what should happen? There should be proofreading and repair. Okay, so in short, in G1S, G2 phases, in G1S, G2 phases, otherwise called as interphase. Why am I calling it as interphase? Because it is interplaced between complete resting phase and active cell division phase. Do you all understand this? In mitochondrial DNA also same, more sensitive to radiation. Yes, mitochondrial DNA, it's almost the same, right? Because it's just that it has got a circular double-stranded DNA. Otherwise, it's going to be the same. It is just that it's done by DNA polymerase gamma in S phase. Otherwise, almost the same. Okay. So, what was I telling you? So, in G1S, G2 phases, it's otherwise called as interphase. Why do we call it as an interphase? Because it is interplaced between complete resting phase and active cell division phase. So, in interphase, continuously there is proofreading and repair or replication happening. So, for proofreading and repair and for replication, what form of chromosome will you want? You want an uncondensed form of chromosome. So, please remember, in interphase, your chromosome is going to be uncondensed, and that is why in interphase, this is how chromosomes would look. Can you see that here? All chromosomes are looking like thin, long threads. Okay. So, to summarize, can you tell me about... So, can you now tell me about phases of cell cycle? Quick recap of all that I've told you. Phases of cell cycle would be, you start with G0 phase, it gets into G1, S, G2 and M phase. G0 is a complete resting phase wherein neither replication nor transcription happens. When it need not perform replication and transcription, to enable package, your chromosome will be highly condensed. Similarly, in M phase, you want your chromosomes to break and segregate, for which you want again condensed form of chromosome. But the difference between a chromosome in G0 phase and a chromosome in M phase is, in M phase, chromosomes have undergone duplication. So, where does duplication happen, all of you? Duplication or replication happens in S phase. Now, what is G1 phase? G1 phase is a pre-synthetic phase where you proofread and repair chromosomes. Very good, all of you. All of you, please start answering. Now, what is G2 phase? G2 phase is a post-synthetic phase where you again proofread and repair. So, G1S, G2 phases together called as interphase, your chromosomes will be uncondensed. Got it? So, that's about this MCQ. So, can you try answering this MCQ now? Which of the following is a true statement? Where does replication happen? You know, this is G0 phase. G0 phase should be followed by A is nothing but G1 phase. B is nothing but S phase. C is G2 phase and then M phase. And we know replication happens in S phase or in B phase. So, replication happens in B phase, but in B phase the chromosomes will be uncondensed. So, what is the right answer here? The right answer here is choice C. Replication happens in B phase. In B phase, the chromosome is uncondensed. Okay. So, the next MCQ. So, this is the next MCQ. Can anybody try answering this? Isochromosome is a defect of which of the following stages? So this is A, B, C, D and E. And wherever mitotic spindle is formed, if mitotic spindle is formed, it's attached to the centromere, what will you call it as? This is called as metaphase. Okay, this is metaphase. And do you see that here mitotic spindle contracts and one half of chromosome goes to one pole, the other half of the chromosome goes to the other pole. So what is E? E is anaphase. Okay, so this is metaphase, this is anaphase and this is telophase. So what are the stages which are shown here? Hi Karthik Narayan. Yes, metaphase and anaphase. Very good. So the question is isochromosome. First tell me what is isochromosome. You know every chromosome is supposed to have a short arm and a long arm. And if there is a defect, because of which if a chromosome gets to have either two short arms or two long arms, that is called as isochromosome. When do you think isochromosome will be the result? During anaphase, when the mitotic spindle contracts, the centromere instead of breaking longitudinally, if it breaks transversely, then two short arms will go to one pole, the other two long arms will go to the other pole. So isochromosome is a defect of which phase? It's a defect of anaphase. Where do you see anaphase here? E is anaphase. Do you understand this? E is anaphase. So what is the right answer? The right answer is choice C. Okay, right answer is choice C. So can we quickly discuss stages of mitosis? Yeah, can we quickly discuss stages of mitosis? First tell me what is mitosis. Mitosis happens in somatic cells. And as a result of mitosis, one diploid cell will give rise to two diploid cells. Don't we know that? Yeah, mitosis happens in somatic cells. As a result of mitosis, one diploid cell will give rise to two diploid cells. Okay. Now, this happens in M phase of cell cycle. Don't we know that? Mitosis happens in M phase of cell cycle. And prior to M phase, you know there will be interphase. Can you tell me what happens in interphase? As soon as the cell decides to get into cell cycle, it first gets into interphase. In interphase, first there is uncondensation of chromosome. Yeah, so now every chromosome will not have a discrete centromere, a short arm and a long arm. Every chromosome will look like a long thread. Okay, uncondensation happens. And then what happens in S phase? In S phase, duplication happens. So following duplication, every chromosome will now get to have two short arms and two long arms. Two long threads which are present close to each other. Such an uncondensed duplicated chromosome of interphase is getting into mitosis. And mitosis has got prophase, metaphase, anaphase and telophase. Don't we know that? Mitosis has got prophase, metaphase, anaphase and telophase. What happens in prophase? Always in prophase there are two events which happen. The first event is condensation of chromosome. Yeah, the first event is condensation of chromosome. So why am I drawing a chromosome with two short arms and two long arms here? To tell you that the chromosomes have been duplicated already. So what is the first event that happens in any prophase? In all the prophases, the first event that happens is condensation of chromosome. The second event that happens in every prophase is pairing of homologous chromosomes. What do you mean by homologous chromosomes? You know that we have two copies of every chromosome, right? Yeah, you know that we have two copies of every chromosome. Chromosome 1, you have paternal copy and maternal copy. So those two copies of every chromosome will be called as homologous chromosomes. So the two events that happen in any prophase will be first condensation of chromosome, second is pairing of homologous chromosomes. After prophase, it gets into metaphase. So tell me what happens in metaphase. Metaphase is always for mitotic spindle formation. So in metaphase, mitotic spindle is formed and it gets attached to the centromere. Yeah, in metaphase, mitotic spindle is formed, it gets attached to the centromere. And then what happens in anaphase? In anaphase, when the mitotic spindle contracts, the centromere breaks longitudinally, so that one short arm and one long arm goes to one pole, the other short arm and the other long arm goes to the other pole. So tell me what happens in anaphase. In anaphase, when the mitotic spindle contracts, the centromere breaks longitudinally. One short arm and one long arm of one chromosome goes to one pole, the other half goes to the other pole. And in telophase, nuclear membrane reappears. Okay, so these are the events which happen. Very good. Yes, mitotic spindle is formed, 2 copies. Very good. Excellent. So these are the events that happen in mitosis. So when will isochromosome be the result? Isochromosome is the result of anaphase, wherein when the mitotic spindle contracts, the centromere, instead of breaking longitudinally, if it breaks transversely, 2 long arms will go to one pole and 2 short arms will go to the other pole. Okay, so tell me what is the right answer for this question. The right answer is isochromosome is a defect of anaphase. Anaphase is E. Okay. So this is the right answer. Very good. Excellent, Swapnil. So that's all about image-based MCQs in genetics. Let's now discuss a few metabolism-based MCQs. Okay. So here in this MCQ, they've asked you to identify A. And the choices which are provided include fructose 1,6-bisphosphate, fructose 2,6-bisphosphate, phosphofructokinase 1 and phosphofructokinase 2. What is A in the image? This is A. Okay. And in this image, do you see that there is already phosphofructokinase 1? Yeah, there is already phosphofructokinase 1. There is already phosphofructokinase 2. Okay. And you also see fructose 1,6-bisphosphate. Yeah, you already see fructose 1,6-bisphosphate. So, of all these choices, you see everything. You see PFK1, you see PFK2, you also see fructose 1,6-bisphosphate. So, what are you left with? This is why you should not be worried when you see an image-based MCQ. Yeah, whenever there is an image-based MCQ, many a times the clue will be there in the image. All that is required is you have to be mindful, look at the image, try to get clues out of it. So, what is the right answer here? Yeah, it's a no-brainer. You're right, Swapnil. So, what is the right answer here? The right answer here is fructose 2,6-bisphosphate. But let's not take chances like this. Yeah, let's not take chances like this. We should understand what is the significance of phosphofructokinase 1, what is the significance of phosphofructokinase 2, what does fructose 2,6-bisphosphate do? Can we learn about that? Yes? Yeah. If you want to learn about that, can I see a thumbs up from all of you? Yeah. So now I'm going to tell you something complex as far as metabolism is concerned. So first, thank you, Shalini. Thank you for saying yes. Unloading oxygen is not done by fructose 2,6-bisphosphate, Dinesh. Unloading of oxygen is done by 2,3-bisphosphoglycerate. That is totally different. Yeah, 2,3-bisphosphoglycerate reduces the affinity of hemoglobin for oxygen, helps in unloading oxygen to tissues. But here it is fructose 2,6-bisphosphate. So let me start from the very basic fact that you need to know. First, phosphofructokinase 1 is the rate limiting enzyme of what? Phosphofructokinase 1 is the rate limiting enzyme of glycolysis. So what is the rate limiting enzyme of gluconeogenesis? Something which reverses phosphofructokinase 1 activity. And it is fructose 1,6-bisphosphatase. Are you clear about these two facts? Yeah. Now about regulation of glycolysis and gluconeogenesis. So this image is all about regulation of glycolysis and gluconeogenesis. I'm going to tell you all the concepts that you need to know here. So as far as glycolysis and gluconeogenesis are concerned, because both these are reversal of each other, you will have to regulate both these pathways in such a way that when one pathway is active, the other one has to be inactive. Otherwise, they will end up in futile cycles or waste cycles. Yes or no? Yeah, because glycolysis and gluconeogenesis are reversal of each other. You will have to regulate these two pathways in such a way that when one pathway is active, the other one has to be inactive. Otherwise, it will end up in futile cycles. Let me give you an example. Yeah, imagine a person who is in starvation. Yeah, imagine a person who is in starvation and this person has switched on his or her gluconeogenesis, and this person has come up from 2 molecules of pyruvate Yeah, the person has switched on gluconeogenesis, and this person has come up from 2 molecules of pyruvate till 1 molecule of fructose 6-phosphate. Yeah, till 1 molecule of fructose 6-phosphate. In this scenario, tell me what will happen if you have not inhibited glycolysis. If you fail to inhibit glycolysis, then phosphofructokinase 1 will start acting on fructose 6-phosphate, will convert it into fructose 1,6-bisphosphate. And this starts getting into glycolysis. Won't you call it as a waste cycle? Because it has started getting into glycolysis, won't you call it as a waste cycle? Because the person has ended up using 11 high energy phosphates. All of you please remember, for gluconeogenesis to happen, you need 11 high energy phosphates. So here the person has invested 11 high energy phosphates, and the person has come up from 2 molecules of pyruvate till fructose 6-phosphate. But because you failed to inhibit glycolysis, it has started getting into glycolysis. This is a waste cycle because the person will not get what he or she wants. The person wants glucose. That is not happening here. That is why you should regulate both these pathways in such a way that when one is active, the other one has to be inactive. When do you think such a regulation is possible? Such a regulation is possible only when you have a common allosteric regulator. And that common allosteric regulator should stimulate one pathway and it should inhibit the other pathway. Isn't that logical? Yeah, you can regulate these 2 pathways that way. Only when you have a common allosteric regulator which stimulates one pathway, it should inhibit the other pathway. And that common allosteric regulator is fructose 2,6-bisphosphate. So I am going to tell you few facts now. Yeah, I want you to memorize those 4 facts. First, listen to what I am saying and memorize these 4 facts, and then I will show you the image to help you understand how this works. Okay, so what is the first fact? The first fact is the common allosteric regulator is fructose 2,6-bisphosphate. Second fact is this fructose 2,6-bisphosphate is a stimulator of glycolysis. What do I mean by that? When I say fructose 2,6-bisphosphate is a stimulator of glycolysis, I mean to say that it stimulates phosphofructokinase 1. The rate limiting enzyme of glycolysis is stimulated by fructose 2,6-bisphosphate. So that's the second fact. When I say it stimulates glycolysis, what is implied? It is implied that it inhibits gluconeogenesis. When I say it inhibits gluconeogenesis, I mean to say that it inhibits fructose 1,6-bisphosphatase. Do you all understand this? When I say it inhibits gluconeogenesis, I mean to say it inhibits fructose 1,6-bisphosphatase. So are you clear about this so far? Yeah, if you're clear about this so far, can I see a thumbs up please? So what are the first few facts that I've told you? The first 3 facts that I've told you is the common allosteric regulator is fructose 2,6-bisphosphate, and this fructose 2,6-bisphosphate is a stimulator of glycolysis, or in other words, it's an inhibitor of gluconeogenesis. Okay. Now the third fact is this fructose 2,6-bisphosphate is a product of a tandem enzyme. Fructose 2,6-bisphosphate is a product of a tandem enzyme by name PFK2. Yeah, so can you see that here? It's a product of a tandem enzyme by name PFK2. So please remember, PFK1 is the rate limiting enzyme of glycolysis. PFK2 is the tandem enzyme which regulates both glycolysis and gluconeogenesis simultaneously. First, what is a tandem enzyme? Tandem enzyme is a bifunctional enzyme. That is, it is one protein. Tandem enzyme is one protein with 2 exactly opposite enzymatic activities. One enzymatic activity will synthesize a compound. The other enzymatic activity will catabolize the same compound. Do you understand this? One enzymatic activity will synthesize a compound. Karthik Narayan, have you understood it? What is a tandem enzyme? Tandem enzyme is a bifunctional enzyme. That is, it is one enzyme with 2 exactly opposite enzymatic activities. One enzymatic activity will synthesize a compound. The other enzymatic activity will catabolize the same compound. And one enzymatic activity will be expressed when the protein is phosphorylated. The other enzymatic activity will be expressed when the same protein is dephosphorylated. Have you all understood what is a tandem enzyme? An example of tandem enzyme is PFK2. I call it as a tandem enzyme because though the name is PFK2, it has got both PFK2 activity and fructose 2,6-bisphosphatase activity. Yeah. Why am I calling it as a tandem enzyme? Because though I call it as PFK2, this PFK2 has got both PFK2 activity. What is PFK2 activity? It converts fructose 6-phosphate to fructose 2,6-bisphosphate. But it also has got fructose 2,6-bisphosphatase activity, which will cleave fructose 2,6-bisphosphate back to fructose 6-phosphate. Okay, and the last fact is when this tandem enzyme gets phosphorylated, it will act as fructose 2,6-bisphosphatase. When this tandem enzyme gets phosphorylated, what will it act as? When this tandem enzyme gets phosphorylated, it will act as fructose 2,6-bisphosphatase. So what is implied? When this tandem enzyme gets dephosphorylated, it will act as PFK2. When it is dephosphorylated, it will act as PFK2. So can you all answer along with me the 4 facts? Yeah, please tell me the 4 facts that I've told you so far. What is the first fact I told you? The first fact is this: the common allosteric regulator which regulates glycolysis and gluconeogenesis is fructose 2,6-bisphosphate. Second fact, fructose 2,6-bisphosphate is a stimulator of glycolysis, or in other words, it's a stimulator of PFK1. So it's implied that it's an inhibitor of gluconeogenesis. It inhibits fructose 1,6-bisphosphatase. Okay, that's the second fact. Now the third fact is fructose 2,6-bisphosphate is a product of a tandem enzyme by name PFK2. Though you call it as PFK2, it has got both PFK2 activity and fructose 2,6-bisphosphatase activity. The last fact is when this tandem enzyme gets phosphorylated, it will act as fructose 2,6-bisphosphatase. So what is implied? When it gets dephosphorylated, it will act as PFK2. Got it? Yeah. So have a look at this image now. So all that I told you so far is seen in this image. Okay. So what is the first fact I told you? The first fact I told you is this, which is the common allosteric regulator fructose 2,6-bisphosphate. What does it do? It stimulates glycolysis and it inhibits gluconeogenesis. And this fructose 2,6-bisphosphate is a product of a tandem enzyme by name PFK2. Though we call it as PFK2, it has got both PFK2 activity and fructose 2,6-bisphosphatase activity. PFK2 activity converts fructose 6-phosphate to fructose 2,6-bisphosphate, and fructose 2,6-bisphosphatase will cleave all the fructose 2,6-bisphosphate back to fructose 6-phosphate. Yeah, what is the last fact I told you? When this tandem enzyme gets phosphorylated, do you see that a kinase phosphorylates a tandem enzyme? So when this tandem enzyme gets phosphorylated, it will act as fructose 2,6-bisphosphatase. Whereas when this tandem enzyme gets dephosphorylated, do you see that a phosphatase removes phosphate group? When it gets dephosphorylated, it will act as PFK2. Okay. Now tell me what happens in starvation. PFK2 under the influence of, I'll tell you, Dinesh. So tell me what happens in starvation. I want you to answer. In starvation, what happens to blood glucose level? Blood glucose level decreases. When the glucose level decreases, which hormone will be released into circulation? All of you please start answering. When the glucose level decreases, which hormone is released into circulation? Glucagon. And glucagon acts through GS receptor. S stands for stimulating adenylyl cyclase. So cyclic AMP is produced. So in starvation, what is produced, all of you? In starvation, cyclic AMP is produced. This cyclic AMP activates protein kinase. And this protein kinase phosphorylates tandem enzyme. And once phosphorylated, what will the tandem enzyme act as? It acts as fructose 2,6-bisphosphatase. So in starvation, fructose 2,6-bisphosphatase is stimulated, and this fructose 2,6-bisphosphatase cleaves all the fructose 2,6-bisphosphate. Now that there is no fructose 2,6-bisphosphate to stimulate glycolysis, glycolysis stops. And now that there is no fructose 2,6-bisphosphate to inhibit gluconeogenesis, gluconeogenesis is stimulated and what is produced? Glucose is produced. Do you understand this? Gluconeogenesis is stimulated and glucose is produced. Isn't that what you want in hypoglycemia? Isn't that what you want in starvation? So tell me what happens in starvation. In starvation, glucagon is released. Glucagon acts through GS receptor, which increases cyclic AMP, activates protein kinase. This protein kinase phosphorylates tandem enzyme. Once phosphorylated, tandem enzyme will act as fructose 2,6-bisphosphatase. So this fructose 2,6-bisphosphatase cleaves all the fructose 2,6-bisphosphate. Now that there is no fructose 2,6-bisphosphate to stimulate glycolysis, glycolysis stops and gluconeogenesis is stimulated and glucose is produced. Got it? Yeah, so that's all about this image, and this can be asked as an image based MCQ or it can be asked as a conceptual MCQ. Okay, so tell me what is A here. A is something which stimulates glycolysis. A is something which inhibits gluconeogenesis. What can do that? It is fructose 2,6-bisphosphate. Okay. Answer is B. So A is inhibited by PFK2 is in cytoplasm. Yes, Dinesh Kumar, PFK2 is in cytoplasm. Okay, because both glycolysis and gluconeogenesis, namely, in the sense PFK1 and fructose 1,6-bisphosphatase, both are present in cytoplasm. So something which regulates both those enzymes should also be there in cytoplasm. Okay, so what is A? What does A do here? This is an inhibitor which goes and inhibits here. And what does it inhibit? It inhibits ATP-ADP translocator of mitochondrial membrane. This translocator, as you can see here, takes out ATP, gives in ADP into the mitochondria. So that translocator is inhibited by A. What is it? That is atractyloside. Very good, all of you. I am happy that most of you have answered it right. Shruzana has answered it as A, oligomycin. Oligomycin acts by inhibiting FO component of ATP synthase complex. Don't you know that, Shruzana? Yeah, oligomycin inhibits FO. O stands for oligomycin, right? So this is not oligomycin. What is the right answer? Atractyloside inhibits ATP-ADP translocator. What about amobarbitone? Amobarbitone starts with which alphabet? A. Yeah, first alphabet. So amobarbitone inhibits electron transport between complex 1 and Q. Amobarbitone inhibits electron transport between complex 1 and Q. Now what is nalinomycin? 2,4-dinitrophenol. Very good, all of you. Excellent. No problem, Shruzana. It's all about learning. So what was I telling you? Nalinomycin. So 2,4-dinitrophenol, valinomycin, nalinomycin, nijaricin. All these are uncouplers. Do you understand this? What is nalinomycin? Nalinomycin is an uncoupler. Okay, so what is the right answer here? The right answer here is atractyloside. Now I am going to use this chance to help you remember and recollect about electron transport chain inhibitors. Can we learn that? Yeah, electron transport chain inhibitors. So for you to learn about electron transport chain inhibitors, you should first know complexes of electron transport chain. Yeah, I'm going to tell you the list, Brinda. So what are the complexes of electron transport chain? First, NADH enters into electron transport chain through complex 1. So first tell me, how many stationary complexes do we have? We have 5 stationary complexes, right? What are the 5 stationary complexes? Complex 1, which is NADH linked dehydrogenase. Complex 1 is NADH linked dehydrogenase. That is, electrons from NADH enter into electron transport chain through complex 1. Okay, and then you have complex 2, which is succinate dehydrogenase of citric acid cycle. Complex 2 is succinate dehydrogenase of citric acid cycle. And then you have complex 3, which is cytochrome B and C1. Complex 3 is cytochrome B and C1. And then you have complex 4, which is cytochrome A and A3. Complex 4 is cytochrome A and A3, and I'm not going to show complex 5 here because complex 5 is ATP synthase complex, which cannot be shown as to participate in electron transport. So these are the stationary complexes. Complex 1, 2, 3, 4 and complex 5 is ATP synthase complex. Now what are the mobile complexes you have? We have two mobile complexes. One is ubiquinone or Q. Yeah, one is ubiquinone or Q placed between 1 and 3. The other one is cytochrome C placed between 3 and 4. The next one is cytochrome C, which is placed between 3 and 4. So these are the stationary and mobile complexes which are present in electron transport chain. Now what is the direction of transport of electron? If it's an electron which comes from NADH, it enters into electron transport chain through complex 1. From complex 1, electrons go to Q, right? From complex 1, electrons go to Q. From Q, electrons go to complex 3. From 3, they go to C. From C, it goes to 4. And from 4, what is the final acceptor of electron? From 4, the final acceptor of electron is oxygen. Yeah, please remember, in electron transport chain, the final acceptor of electron is oxygen. So this is for NADH electron. If it's an FADH2 electron, it enters into electron transport chain either through complex 2 or it directly gets into Q. So from complex 2, electrons go to Q. From here we know the direction, right? From Q to 3, 3 to C, C to 4 and from 4 to oxygen. So this is the direction of transport of electron. Now what are the inhibitors which act at various levels? So electron transport between complex 1 and Q here. Yeah, electron transport between complex 1 and Q is inhibited by number 1 is amobarbitone. Yeah, just now I told you, right? It starts with A. I don't know why this pen does not work. So what is the first one? The first one is amobarbitone, starts with A. The next one is pyrisidine A, ends with A. And the third one is rotenone. Yeah, rotenone sounds like vitamin A, right? Yeah, it's not vitamin A, but it sounds like vitamin A. I want you to remember that inhibitors which inhibit electron transport between complex 1 and Q, they are all related to A. How are they related to A? Amobarbitone starts with A, pyrisidine A ends with A, and rotenone sounds like vitamin A. Okay. Now complex 2 or succinate dehydrogenase. Complex 2 or succinate dehydrogenase is inhibited by malonate. Yeah, it is inhibited by malonate. And then electron transport between 2 and Q. Between 2 and Q is inhibited by 2 starts with T. Right? 2 starts with T. It is inhibited by TTFA and carboxyl. Okay, between 2 and Q is inhibited by TTFA and carboxyl. And then electron transport between 3 and C. Between 3 and C is inhibited by BAL and antimycin. How do you remember it? BAL has got 3 letters, right? So between 3 and C is inhibited by BAL and antimycin. Please don't get confused here. This antimycin also starts with the letter A, but it has got nothing to do with complex 1. Yeah, so between 3 and C is inhibited by BAL and antimycin. Now finally, electron transport between complex 4 and oxygen. Between 4 and oxygen is inhibited by common poisons. What are the common poisons? Hydrogen sulfide, cyanide and carbon monoxide. Okay, hydrogen sulfide, cyanide and carbon monoxide. So have you understood this list? Yeah, I'm quickly going to tell you about this. So between 1 and Q is inhibited by amobarbitone, pyrisidine A and rotenone. Complex 2 or succinate dehydrogenase is inhibited by malonate. Between 2 and Q is inhibited by 2 starts with T. So between 2 and Q is inhibited by TTFA and carboxyl. Between 3 and C is inhibited by a 3-letter inhibitor. What is it? BAL and antimycin. Between 4 and oxygen is inhibited by hydrogen sulfide, cyanide and carbon monoxide. And apart from all these inhibitors, you need to know uncouplers. Right? So what are the uncouplers you know? We have physiological uncouplers and artificial uncouplers. Physiological uncouplers include thyroxine, right? Thyroxine and brown adipose tissue. Artificial uncouplers include 2,4-dinitrophenol, valinomycin, nalinomycin and nigericin. All these cause, yes Dinesh Kumar, perfectly right. Yes, all these cause histotoxic hypoxia. Because how much ever oxygen is available, it will not allow. In the presence of these inhibitors, oxygen will not be used by tissues. Because the only process for which oxygen is used by tissues is electron transport chain. Okay, so in the presence of all these inhibitors, which type of hypoxia do you expect? It is histotoxic hypoxia. Okay, what is shuttle? Okay, I will tell you about shuttle now. After this MCQ, I'll tell you about shuttle. Okay, anything else you want to know? Okay, so I told you about physiological uncouplers. Physiological uncoupler includes thyroxine and brown adipose tissue. Artificial uncouplers include 2,4-dinitrophenol, valinomycin, nalinomycin and nigericin. And then oligomycin. Please remember, the names might sound similar, but oligomycin is totally different. How does oligomycin act? O inhibits FO component of ATP synthase complex. And then atractyloside inhibits. Atractyloside starts with AT, right? So have it as a clue. Atractyloside inhibits ATP ADP translocator of the mitochondrial membrane. Okay. So Shruzana wants to know about shuttles. Let me tell you about that. So what is answer here? A inhibits ATP ADP translocator. So it is atractyloside. Okay. Do I tell you about shuttle before discussing this MCQ or after this MCQ? You want to know uncoupler's mechanism of action. Okay. So I think I'll probably discuss all of that now, and then we'll discuss about this image. Okay, or probably in the next session I'll tell you about this image. So first about shuttle. So in glycolysis, so do you understand that electron transport chain complexes are present along the inner side of inner mitochondrial membrane? That you know, right? All electron transport chain complexes are present inside the mitochondria. But is there a source of NADH in cytoplasm? Yes. What is that source? Glycolysis. So you know, in glycolysis, for every molecule of glucose that gets into glycolysis, you get 2 NADH. Don't you know that? Yeah, in glycolysis, for every molecule of glucose that gets into glycolysis, you get 2 NADH in the cytoplasm. And these 2 NADH electrons should enter into mitochondria. But as NADH, they cannot cross the mitochondrial membrane. That is why we need a shuttle. Okay, and there are 2 shuttles. One is a malate shuttle, the other one is a glycerol phosphate shuttle. So first let me tell you about malate shuttle. When I say malate shuttle, I mean to say, in the case of malate shuttle, you transport electrons in the form of malate. So when you call it as malate shuttle, what does it mean? It means here you transport electrons in the form of malate. And for this we use 2 enzymes. One is cytoplasmic malate dehydrogenase. Yeah, the first enzyme is cytoplasmic malate dehydrogenase. The other one is mitochondrial malate dehydrogenase. Cytoplasmic malate dehydrogenase acts on the NADH which you have obtained through glycolysis. Yeah, cytoplasmic NADH acts on the NADH which you have got through glycolysis. In NADH, are you worried about the niacin part or are you worried about the electron part? We want only the electron. So this cytoplasmic malate dehydrogenase gets the electron from NADH, uses that electron to reduce its substrate, and its substrate is oxaloacetate. So oxaloacetate accepts the electron from NADH, gets converted to malate. So tell me, where is the electron now? The electron is now within malate. Malate carrying the electron crosses the mitochondrial membrane, gets into the matrix. And once malate enters into the matrix, mitochondrial malate dehydrogenase acts on this malate, converts it into oxaloacetate. And during this conversion, electron is taken out of malate and that electron is given to its coenzyme. And because its coenzyme is NAD, this NAD accepts the electron and it becomes NADH. Then this NADH enters into electron transport chain. So now do you see that this way you have translated cytoplasmic NADH as mitochondrial NADH? Shruzana, have you understood this? This way you have translated, I won't say you have transported. What have you done? You have translated cytoplasmic NADH as mitochondrial NADH that then gets into electron transport chain. So this is the major function of malate shuttle. Have you all understood this? So what is the major function of malate shuttle? Malate shuttle helps in transporting or helps in translating cytoplasmic NADH as mitochondrial NADH so that the two NADH which we get through glycolysis will be able to enter into electron transport chain. Okay, and there is there is another shuttle which is glycerol phosphate shuttle. All cells, all tissues have malate shuttle. Yeah, all tissues have malate shuttle. Exception will be white muscle fibers and neurons. Only in white muscle fibers and neurons you don't have malate shuttle. Instead you have glycerol phosphate shuttle. So in glycerol phosphate shuttle, you transport electrons in the form of glycerol 3-phosphate. And for that you use again two enzymes. What are the two enzymes? The first enzyme is cytoplasmic glycerol 3-phosphate dehydrogenase. The next enzyme is mitochondrial glycerol 3-phosphate dehydrogenase. So what is the only difference here? The names, right? The names. So in malate shuttle it is cytoplasmic malate dehydrogenase and mitochondrial malate dehydrogenase. In glycerol phosphate shuttle it is cytoplasmic glycerol 3-phosphate dehydrogenase and mitochondrial glycerol 3-phosphate dehydrogenase. Otherwise it's almost the same story. What does cytoplasmic glycerol 3-phosphate dehydrogenase do? It acts on the NADH which we have got through glycolysis. In NADH you want only the electron. So this enzyme takes the electron out of NADH, gives that electron to its substrate. And because its substrate is dihydroxyacetone phosphate, it accepts the electron and it gets converted to glycerol 3-phosphate. Now the electron is hidden in the form of glycerol 3-phosphate. Carrying that electron, glycerol 3-phosphate crosses the mitochondrial membrane and it gets into the matrix. And within matrix, glycerol 3-phosphate, mitochondrial glycerol 3-phosphate dehydrogenase acts on this glycerol 3-phosphate and converts it into dihydroxyacetone phosphate. It gets converted to dihydroxyacetone phosphate. During this conversion, electron is taken out. When that electron is given to its coenzyme, because mitochondrial glycerol 3-phosphate dehydrogenase uses only FAD as its coenzyme. Mitochondrial glycerol 3-phosphate dehydrogenase uses FAD as its coenzyme. Here FAD gets converted to FADH2. So can you tell me what is the limitation of a cell using glycerol phosphate shuttle instead of malate shuttle? If it's malate shuttle, NADH in the cytoplasm becomes NADH in mitochondria. So there is no change in energetics. But if it's glycerol phosphate shuttle, every cytoplasmic NADH that is getting transported this way will be converted to mitochondrial FADH2. Had it been NADH, it would have given you 2.5 ATPs. Because it is converted to FADH2, you are going to get only 1.5 ATP. So for every NADH electron that is getting transported this way, you are going to miss 1 ATP. Do you all understand this? No, this is not about faster ATP production, Brinda. This is about difference in the ATP that gets produced. So for every NADH electron that is getting transported this way, you are going to miss 1 ATP. From every molecule of glucose, you get 2 NADH. So if a cell uses glycerol phosphate shuttle, when those 2 NADH become 2 FADH2, we are going to miss 2 ATPs. Yeah, we are going to miss 2 ATPs. So if they specifically give you that a cell uses glycerol phosphate shuttle and they are asking you for number of ATPs produced, whatever number that you have memorized so far, you should subtract 2. Do you understand this? If they specify that a cell uses glycerol phosphate shuttle, whatever number of ATP that you have memorized so far, you subtract 2. For example, glucose on aerobic glycolysis gives you how many ATPs? I've given you the answer. Glucose on aerobic glycolysis gives you 7 ATPs. But if a cell uses glycerol phosphate shuttle, how many ATPs will you get? If it's glycerol phosphate shuttle, you can get only 5 ATPs. Glucose on complete oxidation gives you 32 ATPs. Right, glucose on complete oxidation gives you 32 ATPs. But if a cell uses glycerol phosphate shuttle, it becomes 30. Okay, anaerobic glycolysis, from every molecule of glucose you get 2 ATPs. And if a cell uses glycerol phosphate shuttle, how many ATPs do you expect? Can anybody answer this? Yeah, 32, 30, other than that. Anaerobic glycolysis usually gives you 2 ATPs. If a cell uses glycerol phosphate shuttle, it is going to be still 2. Why will it be still 2? Because for anaerobic glycolysis, will you need electron transport chain at all? For anaerobic glycolysis, will you need electron transport chain at all? No. So they might try to confuse you. They might say one molecule of glucose enters into anaerobic glycolysis in a cell which uses glycerol phosphate shuttle. How many ATPs will you get? Irrespective of the shuttle that is present in a cell, anaerobic glycolysis always gives you 2 ATPs. Okay, yes, electron transport chain does not even happen. Okay, so that's about malate shuttle and glycerol phosphate shuttle. Have you understood this? And what other fact did you ask me? I think one other person was asking me about mechanism of uncoupler, right? Who was asking me? Dinesh was asking about uncoupler's mechanism of action. So tell me about how electron transport chain works. So in electron transport chain, whenever, one second. So in electron transport chain, when electrons go through all these complexes, in electron transport chain, when electrons go through all these complexes, what happens? So in electron transport chain, when electrons go through all these complexes, they move from a complex of higher energy to a complex of lower energy. And that liberates energy. And the energy that is liberated is used for pumping hydrogen ions from the matrix to outside the inner mitochondrial membrane. So what I have drawn here as a red structure, this is nothing but inner mitochondrial membrane together. Okay, and where are these complexes arranged? We know that all these electron transport chain complexes are arranged along the inner side of inner mitochondrial membrane. And when electrons move from one complex to another, energy gets liberated. And the energy that is liberated is used for pumping hydrogen ions from the matrix to just outside the inner mitochondrial membrane. Do you understand this image? When electrons go through electron transport chain complexes, energy is liberated, and that energy is used for pumping hydrogen ions from the matrix to just outside the inner mitochondrial membrane. And after accumulating hydrogen ions along the outside of the inner mitochondrial membrane, these hydrogen ions find their way through FO component of ATP synthase complex. I said complex 5 is ATP synthase complex, and this is complex 5. This complex 5 has got 2 subunits. One is FO subunit, which is an ionophore through which hydrogen ions can go. The other one is F1 subunit, which is the actual ATP synthase enzyme. So after hydrogen ions accumulate outside the inner mitochondrial membrane, these hydrogen ions find their way through FO component of ATP synthase complex. And here it's a movement of hydrogen ion from a region of higher concentration to a region of lower concentration. And that liberates energy. That energy is finally used for phosphorylating ADP to form ATP. Do you all understand this? This is how you generate ATP with the help of electron transport chain. Now what is an uncoupler? Uncoupler uncouples oxidation from phosphorylation. First tell me, what does electron transport chain do? Electron transport chain couples oxidation of fuel with phosphorylation of ADP to form ATP. Yes or no? What is the function of electron transport chain? This is electron transport chain, and this electron transport chain couples oxidation of fuel with phosphorylation of ADP to form ATP. Now, what do you think an uncoupler does? An uncoupler uncouples oxidation from phosphorylation. Yeah, uncoupler uncouples oxidation from phosphorylation in the sense oxidation of fuel happens continuously without phosphorylation. Yeah, oxidation of fuel happens continuously without phosphorylation. So, what are the effects of uncouplers? When there is no phosphorylation, the first effect is no ATP production. Yeah, the first effect is no ATP production, but still oxidation of fuel is happening continuously. Continuously energy is getting liberated. You are not trapping the energy as ATP. If you don't trap the energy, energy will be liberated out as heat. So, the second effect of uncoupler is because oxidation of fuel is happening continuously, energy will be liberated out as heat. So, there is increase in the rate of heat production. When there is no ATP production, for want of ATP, more and more fuels will get oxidized. So, there will be an increase in the rate of oxidation of all the fuels. Yeah, there will be an increase in the rate of oxidation of all the fuels. So, these are the effects of uncouplers. Do you all understand this? Can you make a schedule to revise biochem? Yeah, I will do it. Yeah, this is a part of the revision series. I will definitely do it. Okay. So, tell me, what are the effects of uncouplers? There are three effects of uncouplers. The first effect is no phosphorylation. So, no ATP production. Still oxidation of fuel is happening continuously and energy is getting liberated out as heat. So, the second effect is increase in heat production. When there is no ATP production, for want of ATP, more and more fuels will be oxidized. So, there is increase in the rate of oxidation of all the fuels. So, these are the effects of uncouplers. But how do they bring about these effects? Because these uncouplers are ionophores. All these uncouplers are ionophores, or they are ion channels. And these ion channels, let me show uncouplers action in blue color. So, all these uncouplers are ionophores or ion channels, and these ion channels get inserted into the inner mitochondrial membrane. And through these ionophores or ion channels, hydrogen ions will bypass. And because hydrogen ions do not go through FO component of ATP synthase complex, ATP will not be produced. But even here, hydrogen ions move from a region of higher concentration to a region of lower concentration. So, energy will be liberated out as heat. Do you understand the mechanism of action? It's very simple. All ionophores are ion channels. All uncouplers are ionophores or ion channels, and they get inserted into the inner mitochondrial membrane. Through these ionophores or ion channels, because hydrogen ions bypass, there is no ATP production, but there is only heat production. Okay. An example for these uncouplers will be physiological uncouplers and artificial uncouplers. Physiological uncouplers will be thyroxine. I hope you must have learnt that thyroxine increases the basal metabolic rate. Why do you say it always increases the basal metabolic rate? Because it causes insertion of uncouplers into the inner mitochondrial membrane. So, hydrogen ions keep getting bypassed, energy is getting liberated out as heat. For want of energy, more and more fuels will be oxidized. That is why we say it increases the rate of oxidation of all the fuels. Okay, the other one is brown adipose tissue. I hope it's clear. Okay, and all these artificial uncouplers like 2,4-dinitrophenol, valinomycin, nalinomycin, nejaricin, all these were once tried as anti-obesity drugs. Will you agree with me if I say uncouplers can be tried as an anti-obesity drugs? True, right? Because they increase the rate of oxidation of all the fuels without ATP production, only heat generation. So, any fuel that has been stored by the individual will be oxidized and only heat gets generated, no anabolism. So, it can help in weight loss. So, 2,4-dinitrophenol, valinomycin, nalinomycin, nejaricin, all these were once tried as anti-obesity drugs. But because they all presented with posterior subcapsular cataract, and because they all presented with increased heat production, yeah, intolerable heat production, they were all withdrawn from market. Okay, so I hope this helped. So that's all about, yeah, so that's all for today's session. I will see you soon with another session. Okay, so thank you. Good night.